Hello, my name is Ellie Sempek. I am a second-year biochemistry and molecular biology student working in Dr. Karen Helt's bioseparations laboratory. The project discussed today is the analysis of the hydrophobicity of viruses using absorption isotherms. The goal of the project is to analyze the hydrophobicity of porcine parvovirus, or PPV, to optimize the virus purification process, improve chromatographic resin selection, and measure the virus's hydrophobicity. This experiment gives important information on virus purification processes, which are necessary to meet FDA guidelines and maintain good manufacturing practices. The virus analyzed in this study is a model virus, which gives general information on a virus with a specific property. In this case, we tested an enveloped virus, PPV. The surface chemistry of a virus depends on the presence of this envelope and ultimately affects the purification process, which is necessary in order to remove proteins and other impurities from the virus, so it can be used for manufacturing. Analyzing absorption isotherms allows us to observe the effects of different solution conditions on virus absorption. Absorption isotherms are a graphical representation of the difference in absorption between materials that can be compared to view the extent of absorption. We used hydrophobic chromatography resins, phenyl and butyl sulfurose resins, as absorbents, while the absorbate it used is PPV at different viral and salt concentrations. In this experiment, the virus is diluted with phosphate-buffered saline solution at different salt concentrations. This is to test the effect of salt concentration on the absorption of PPV to the resins. The expected effect is that it leads to an increase in binding between the resins and the virus. To obtain the difference between absorbed and unabsorbed virus onto the resins, an MTTSA is performed. The MTTSA is a colorimetric method based on the determination of cell viability. It measures the concentration of virus left unabsorbed after mixing with the resins and allows the hydrophobicity of the virus to be tested. The virus is loaded into a tube containing the hydrophobic resins and mixed thoroughly. This ensures that the resins and virus get the maximum amount of interaction. After mixing, a supernatant is allowed to settle above the resins. This supernatant contains the unabsorbed virus and will be collected for the MTTSA to measure the extent of absorption. This is the experimental data for PPV in 155 millimolar PBS, the lower salt solution. This graph shows that phenyl resins are indeed more hydrophobic than butyl resins, as we saw more virus binding with the phenyl resins. This gives us viable information on the benefits of certain resin types at a lower salt concentration. The experimental data for PPV in 350 millimolar PBS shows that with increasing virus concentration, the adhesion of the virus to the resins increases. It also shows that the increased salt concentration increased the binding abilities of the butyl resin. This means that a more hydrophilic resin could be used with a high salt concentration and still produce good adhesion, which gives viable information on selecting resins and solution conditions. The experimental data is fitted into absorption isotherm graphs of different theoretical models, Langmuir and Freundlich. These models are the standards for analyzing absorption isotherms. Langmuir isotherms are based on the surface of the resins being homogeneous, whereas Freundlich isotherms are based on non-homogeneous surfaces. The model with the best fit was the Freundlich model and is the model used in the graphs. This is because the data points line up with the best with this model fit. A Freundlich best fit tells us that the surface of the hydrophobic resins are both non-homogeneous. These graphs show that PPV is hydrophobic because the absorption isotherm graph showed a sloped line. This slope means the virus was binded to the resins, which only occurs if the virus is hydrophobic. Therefore, the hydrophobicity of PPV was analyzed using absorption isotherms. This data has allowed us to understand the hydrophobicity of viruses better, and our knowledge on how hydrophobicity affects the purification process has improved. The authors would like to thank the Department of Chemistry and the Department of Chemical Engineering at Michigan Technological University and the Helt Bioseparations Laboratory for supporting this work. In the future, this experiment will be run using a non-enveloped virus, bovine viral diarrhea virus, or BVDV. This will allow the hydrophobicity of a virus with a different surface chemistry to be analyzed. Thank you for your time.